Hey everyone, it's Dr. Marcon, and this is the first part of Chapter 4 on tissues. We talked about the hierarchy or levels of organization within an organism the other day. So we know that cells work together in functionally related groups called tissues. So cells make up tissues, and tissue is defined as a group of closely associated cells that perform related functions and are similar in structure. There are four basic tissue types and basic functions. The first tissue type we talked about was epithelial tissue. We went over that in lab. So epithelial tissue, their function is mostly for covering. We can see types of epithelial tissue in chapters 4 and 5. We also have connective tissue. Connective tissue, the main function of this type of tissue is for support. And we talk about connective tissue in chapters 4, 5, 6, and 9. The next type of tissue is muscle tissue, and we know that muscle tissue is mainly responsible for movement. And we talk about muscle tissue in chapters 10 and 11. And the last type of tissue is nervous tissue, and we know that the nervous tissue is responsible for control. So it controls everything about our bodies. So nervous tissue we'll talk about in chapters 12 through 16, as well as chapter 25. So epithelial tissue, its main function is for covering. Epithelial tissue covers a body surface or lines a body cavity. Epithelial tissue also forms parts of most glands. Functions of epithelia include protection, secretion, absorption, diffusion, filtration, and sensory reception. We have special characteristics of epithelia, one of them being cellularity. Cells are separated by minimal extracellular material, and epithelia are composed almost entirely of cells. So we have a lot of cells separated by a small amount of extracellular material. Another characteristic of epithelia is that they have specialized contacts, or a specialized way of communication between cells. Cells are joined by special junctions that allow for communication between cells. Another special characteristic is polarity. We have the cell regions of the apical surface will be different from the basal surface. We know that epithelia are supported by connective tissue. In some mucous membranes, we have a special type of support called the lamina propria. We also know that epithelia are avascular but are innervated. So if you ever see the prefix A usually means without. So avascular means that epithelia do not have blood supply specifically, but they do receive blood supply and nutrients from underlying connective tissue. So that's why the connective tissue be below that layer of epithelia is so important. So epithelia are avascular, meaning they don't really have a blood supply, but they do have nervous innervation, so innervation by nerves. Regeneration is also another special characteristic of epithelia. Lost cells are quickly replaced by cell division. So when we saw the stratified epithelium in lab, we knew that the cells along that basement membrane are very important and are very active in dividing to help replace the dying cells at the top layers of the stratified epithelium. So here we can see some special characteristics of epithelium. We know that this is the apical surface of the cells of the epithelium, and we have our basal region or basement membrane, so the bottom part of the epithelium. If this is only one layer, we know that this is a simple epithelium. We can also see specific junctions between cells that either hold the cells together and also provide communications between cells. So we have different types of cell junctions, which we'll get into. We have tight junctions, we have gap junctions, all very important for communication between cells. So when we talk about epithelium being avascular, meaning we don't have a direct blood supply to the epithelium, rather within the connective tissue below the epithelium, we can see that we have capillaries which provide blood to the epithelium, as well as innervation from nerve endings within the connective tissue that go to the epithelium. When we classify epithelium, we want to start first by looking at specific parameters. For example, first we want to look at the number of layers. So the first name of the epithelial tissue indicates the number of cell layers. If there is only a single layer, we classify this type of epithelium as simple epithelia. Okay, so simple epithelia is a single layer of cells attached to a basement membrane. If there are multiple layers, we then say that the epithelium is stratified. So stratified epithelia have multiple layers of cells, and the basal layer of cells, again, attached to the basement membrane. So first we look at the number of layers. The next part of the name describes the shape of the cells, specifically the cells that are on the apical surface of the epithelium. We consider epithelium to be squamous if the cells look like plates or discs, so cells are wider than they are tall. If the apical cells look like they are cubes, we call them cuboidal, so cells that are wide as tall and look like cubes. And if the apical cells look like columns, we call them columnar because these cells are taller than they are wide and they actually look like columns. And then this next figure shows you a depiction of the different types of epithelium based on shape. So here we have squamous cells that look like discs or plates. We have cuboidal cells that look like cubes, and then we have columnar cells that look like tall columns, so they are taller than they are wide. So again, first we look at the number of layers. If it's only one layer, we call it simple. If there's many layers, we call it stratified, and then we classify the epithelium based on cell shape, so either squamous, cuboidal, or columnar. This table is actually shows the function of epithelial tissue related to tissue type. We know that simple squamous epithelium, their main function is for diffusion and filtration. We saw this type in the alveoli of lungs or the Bowman's capsule in the kidney. Both structures very important for either diffusion of gases or filtration of blood to form urine in the kidney, for example. If we have stratified squamous epithelium, the main function of the stratified squamous epithelium is for protection. We see this in the lining of our organs, internal organs, or even, the stratified squamous layers of our skin. Simple cuboidal and simple columnar epithelium, their main function is for secretion and absorption. There are ciliated types, specifically ciliated columnar types, that can propel mucus or reproductive cells. Stratified types of this type of epithelium, mainly for protection. However, it's very rare to see stratified cuboidal or stratified columnar epithelium. It's not very common. And then we have transitional epithelium. The function of transitional epithelium is more for protection, as well as stretching allows for accommodation of distension of urinary structures, like the urinary bladder. So first we're going to talk about simple squamous epithelium. A description of this type of epithelium, it's a single layer, simple indicating one layer, and squamous, these cells are flat with disc-shaped nuclei. Function of the simple squamous epithelium allows for passage of materials by passive diffusion and filtration. Also can secrete lubricating substances within the serosa. Location of our simple squamous epithelium includes renal corpuscles, specifically the Bowman's capsule or the glomerular capsule surrounding the glomerulus within the kidney, alveoli of the lungs, these air sacs that allow for diffusion of gases, our respiratory gases, oxygen and carbon dioxide. We can also find simple squamous epithelium in the lining of the heart, blood, and lymphatic vessels, and then the lining of ventral body cavities. So just a kind of recap of simple squamous epithelium. Its description, again, being that single layer flattened cells, function allowing for diffusion and filtration, specifically in the lungs and the kidney, And again, they can be located in the glomeruli of the kidney, that Bowman's capsule, the alveoli of the lungs, lining of the heart, blood vessels, and lymphatic vessels, as well as lining of the ventral body cavity. So here is that thin layer of simple squamous epithelium, this specifically within the alveolar walls in the lung. Simple cuboidal epithelium is a single layer of cube-like cells with large spherical central nuclei. Function of our simple cuboidal epithelium includes secretion and absorption, and we can actually find this type of epithelium in kidney tubules, secretory portions of small glands, as well as the surface of the ovary. Again, simple indicates one layer, so we have a single layer of cube-like cells with large spherical central nuclei. Function of this type of epithelium includes secretion and absorption, and we can find them in kidney tubules, ducts, secretory portions of small glands, and ovary surface. And mind you, the kidney is actually a really good place to find not only simple cuboidal epithelium, but also simple squamous epithelium. The next type of simple epithelium is the simple columnar epithelium. So this is a single layer of column-shaped or rectangular cells with oval nuclei. Some can have cilia at their apical surface, and some can contain goblet cells. Goblet cells are cells that secrete mucus onto the apical surface of epithelium. Function of simple columnar epithelium includes absorption, as well as secretion of mucus, enzymes, and other substances. Ciliated type of simple columnar epithelium can propel mucus or reproductive cells by ciliary action. We can find simple columnar epithelium in various places. The non-ciliated form usually is found in the lining of the digestive tract, within the gallbladder, and then the ducts of some glands, whereas the ciliated form of simple columnar epithelium can be found in the lining of small bronchi, fallopian tubes or the uterine tubes, as well as the uterus. So here is actually a really lovely picture of simple columnar epithelium. We can see that the cells are nice, tall columns. We can see that the nuclei are lined up in this lovely line within the cell. So again, simple columnar epithelium made up of a single layer of tall cells with round to oval nuclei. Some can have microvilli or cilia on the apical surface of these cells. Within the epithelium, you may find mucus-secreting unicellular glands called goblet cells that produce mucus. Function of the simple columnar epithelium, again, absorption, secretion of mucus, enzymes, other substances. Again, the ciliated types of simple columnar epithelium can propel mucus or reproductive cells by the action of the cilia. We can find non-ciliated types in the digestive tract from the stomach to the anal canal. We can find it in the gallbladder as well as excretory ducts of some glands. A ciliated type of simple columnar epithelium, again, we can find in the small bronchi, fallopian tubes, and some regions of the uterus. The next type of simple epithelium is pseudo-stratified columnar epithelium. Again, pseudo means fake. So this type of epithelium actually gives the appearance of stratification within this epithelium, or many layers. However, pseudo, because it means fake, that means it's actually only one layer of epithelium. So within pseudo-stratified columnar epithelium, again, the shape of these cells are columnar, meaning they're tall cells. All the cells originate at the basement membrane, but only the taller cells can reach the apical surface. They may contain goblet cells and bear cilia. The nuclei, compared to simple columnar epithelium, the nuclei actually can be at different heights within the cells, which gives that false impression of stratification or of layering of cells. But it's actually, one layer of cells with the nuclei at different levels within the cell. Functions of pseudostratified columnar epithelium, secretion of mucus, as well as propulsion of mucus by cilia. We can find the non-ciliated type in the ducts of male reproductive tubes, as well as the ducts of large glands. And then we can find the ciliated variety lining the trachea and most of the upper respiratory tract. These cells are very important in the upper respiratory tract because not only do they secrete mucus, but they also propel the mucus so that you can actually expectorate this mucus out of the body. For example, if the mucus traps any bacteria or pathogens or any irritants like allergens that we breathe in, these can be expelled from the body by that ciliary motion, which is why if you're a smoker, the smoke that you inhale from cigarettes or from anything else can actually damage the cilia of the pseudostratified columnar epithelium, and you become more prone to infections, such as pneumonia. So the take-home message is, don't smoke cigarettes, kids. Here's a slide that shows the overall general characteristics of pseudostratified columnar epithelium. Again, we have a single layer of cells of different heights. Some cells will not reach the free surface, and we can see that the nuclei are at different levels, giving it that pseudostratified appearance. They may contain mucus-secreting goblet cells and bare cilia at the apical surface. Function of your pseudostratified columnar epithelium includes secretion, particularly of mucus, as well as propulsion of mucus by that ciliary action. We can find the non-ciliated type of pseudostratified columnar epithelium in male sperm-carrying ducts, as well as ducts of very large glands. The ciliated variety lines the trachea, as well as most of the upper respiratory tract. Our next type of epithelia is stratified epithelia. Again, stratified epithelia contains 2 or more layers of cells. This type of epithelia actually regenerate from below, from the cells found at the basal layer. The major role of stratified epithelium is protection, and we know that the stratified epithelia are named according to the shape of cells found at the apical surface or at that apical layer. So the first type we're going to discuss is stratified squamous epithelium. Stratified squamous epithelium has many layers of cells that are squamous in shape. Now, the deeper layers of this epithelium, the cells appear cuboidal or can be columnar, and this is the thickest type of epithelial tissue. We know that is best adapted for protection from abrasion or a wearing down of the topmost layers. We have 2 types of stratified squamous epithelium. We have keratinized and non-keratinized, basically whether or not they contain the protein keratin. So keratinized stratified squamous epithelium is found in the epidermis, which is a layer of skin. Contains the protective protein keratin, basically adds to the waterproof properties of this type of epithelium, so we're not constantly losing water from our bodies. And then we know that the surface cells are dead and full of that protein keratin. Now, the non-keratinized type of the stratified squamous epithelium forms moist lining of body openings. So again, the function of stratified squamous epithelium is to protect underlying tissues in areas that are subject to abrasion. Keratinized stratified squamous epithelium is found in skin. It mostly forms the epidermis. And then the non-keratinized is found lining mucous membranes such as the esophagus, mouth, the anus, the vagina, and the urethra. So again, here is that slide that kind of summarizes the characteristics of stratified squamous epithelium. So it's a thick membrane composed of several cell layers. We know that the basal cells along that basement membrane are either cuboidal or columnar in shape and are metabolically active, meaning they're constantly dividing and kind of replacing the cells that are being lost at the surface. So the surface cells are flattened, meaning they're squamous. They look like plates or discs. In the keratinized type, the surface cells are full of that protein keratin that help with the waterproofing properties, and we know that these cells are dead, whereas the basal cells are active in mitosis and produce the cells of the more superficial layers to kind of replace the cells that are eventually lost. Function of this type of epithelium is to protect underlying tissues in areas that are subjected to abrasion. On the outer surfaces, we have a very thick skin in the palms of our hands and the soles of our feet. Internally, we have our mouths, for example. The epithelium within our mouths are constantly regenerating, especially if we eat something hot or cold. So these cells are constantly sloughed off. So non-keratinized type forms the moist linings of the esophagus, the mouth, and the vagina, whereas the keratinized type forms the epidermis of the skin, which is a dry membrane. So the next type of stratified epithelium is the stratified cuboidal epithelium. Generally, it has only about 2 layers of cube-shaped cells. The main function of the stratified cuboidal epithelium is for protection. We can find this type of epithelium forming the ducts of the mammary glands, the salivary glands, and the largest sweat glands. So again, just a summary of the description of stratified cuboidal epithelium, about 2 layers of cube-like cells. Main function is protection, found in the largest ducts of sweat glands, mammary glands, and salivary glands. So here is actually a picture of a salivary gland duct. We have about 2 layers of cuboidal epithelial cells. Now, stratified columnar epithelium is actually pretty rare. We don't see a lot of this within the body. So it is composed of several layers of columnar cells. We know the basal cells are usually cuboidal in shape, whereas the superficial cells are more elongated and show that column-like shape of the cells. Function of our stratified columnar epithelium includes protection as well as secretion. Again, is a very rare tissue type. We can find this type of epithelium in the male urethra as well as the large ducts of some glands. So here we see a picture of stratified columnar epithelium. We can see the cells at the apical surface looking like tall columns. And then we have usually cuboidal cells at the basement membrane. Again, composed of several layers. Function includes protection as well as secretion and are not very common within the body. So we find small amounts of this type of epithelium in the male urethra and in large ducts of some glands. Now it's transitional epithelium. Transitional epithelium has characteristics of stratified cuboidal and stratified squamous epithelium, depending on whether or not that organ is fully distended or relaxed. So the superficial cells, or the cells at the surface, are dome-shaped when the bladder is relaxed, meaning there's no urine within the bladder, and then they can appear squamous when the bladder is full of urine. Function of this type of epithelium permits distension of the urinary organs when they are filled with urine, and we can find transitional epithelium in the urinary bladder, the ureters, as well as the proximal urethra. So here we see a lovely picture of transitional epithelium. You can see these dome-shaped cells at the surface. We see many layers, but it's called transitional because they can transition from these dome-shaped cells to the more flattened squamous cells, depending on whether or not the urinary organ is full of urine or doesn't have any urine within it. So again, transitional epithelium resembles both stratified squamous and stratified cuboidal. Again, the basal cells are cuboidal or columnar in shape. Surface cells are dome-shaped when there is no urine in the bladder, and then can be squamous-like if the urinary organ is full of urine. So functions include stretching readily and permitting distension of the urinary organ by contained urine, and then again lines the ureters, the bladder, and part of the urethra. So now we're going to get into glands. We have different types of glands. We have endocrine glands and exocrine glands. Now, endocrine glands, note there's an N in endocrine, meaning they have no ducts. Okay, so endocrine glands are ductless glands that will secrete directly into the surrounding tissue fluid. Endocrine glands produce messenger molecules called hormones. And we'll talk more about endocrine glands when we go into endocrinology in chapter 17. But again, the difference between endocrine glands and exocrine glands, endocrine glands have no ducts compared to exocrine glands. And endocrine glands will secrete directly into the surrounding area or the surrounding tissue fluid. Exocrine glands do have ducts. So the ducts will carry products of the exocrine glands to the epithelial surface. And exocrine glands include the following diverse glands. We have mucus-secreting glands, sweat and oil glands, salivary glands, and we have the liver and pancreas. So both the liver and pancreas are glands. Now, when we talk about the pancreas actually has both endocrine and exocrine functions. And we'll talk more about that when we get to endocrinology in chapter 17. And we'll even touch upon that when we get to the accessory organs of the digestive system. Now, goblet cells are unique unicellular exocrine gland. These cells produce mucin, and when combined with water, mucin plus water equals mucus, that very viscous type material that lines the epithelium. So mucin and mucus protect and lubricate many internal body surfaces. Goblet cells are a unicellular, meaning they're one cell. So they're a unicellular exocrine gland. So here's a sort of a picture of a goblet cell. We can see vesicles within the apical surface of the cell that contain mucin, and when mucin is combined with water, produces mucus. So this unicellular exocrine gland will actually secrete mucus to the apical surface of the epithelium. Multicellular exocrine glands have two basic parts. They have an epithelium-walled duct, and they have a secretory unit. We can classify multicellular exocrine glands by the structure of the duct. We can classify them as either simple or compound. Simple exocrine glands do not have any branching, so there are no branches, whereas compound exocrine glands do have branches coming off of the tubes. We can also categorize these types of glands by the secretory unit. The secretory unit can either be tubular, meaning they're in the shape and form tubes. They could be alveolar, in which the secretory cells will form spherical sacs. Or they could be tubulo-alveolar, so a combination of the two. So here we see a figure that shows the different categories and classifications of the multicellular exocrine glands. So we can classify them as either simple or compound. So simple, the duct will not branch. Compound, we can see that there are branches coming from the ducts. So we have a duct here, just a tube forming that duct in the simple duct, and then branches coming off of the duct, which allow them to be categorized as compound ducts. And then we can categorize them according to the structure of the secretory unit. So they can be categorized as alveolar, meaning they are simple, spherical in shape, or they can be tubular, meaning they only have this tube-like structure, or they can be a combination of the two. So we can have compound tubulo-alveolar glands. So we have a combination of tubes plus alveoli. Now we're going to get into lateral surface features of cells, specifically cell junctions. We have factors that actually bind the epithelial cells together. We have adhesion proteins, which can link plasma membranes of adjacent cells. These kind of form the contours of adjacent cell membranes. And then we have special cell junctions. First, we have the tight junction, also known as the zona occludens. These help close off the space between cells, or the intercellular space. Normally, these are found at the apical regions of most epithelial tissue types. Some proteins in the plasma membrane of adjacent cells are fused together. They also help prevent certain molecules from passing between cells of the epithelial tissue. So here we see the tight junctions kind of closing off that intercellular space, preventing molecules from passing through. Then we have adhesive belt junctions, also known as zonula adherens. This is an anchoring type of junction. So we have transmembrane linker proteins that attach to actin microfilaments of the cytoskeleton and bind adjacent cells. With tight junctions, these linker proteins actually form the tight junctional complex around the apical lateral borders of epithelial tissue, so on the sides and at the surface. We then have desmosomes. Desmosomes are main junctions for binding cells together. They are scattered along abutting sides, so sides that are right next to each other, of adjacent cells. The cytoplasmic side of each plasma membrane has what's known as a plaque, and these plaques are joined by linker proteins. We have intermediate filaments that will extend across the cytoplasm and anchor at desmosomes on the opposite side of the cell. And these structures are common in cardiac muscle as well as epithelial tissue. So here we see the desmosomes. We can see the plaques on the cytoplasm side. We can see intermediate filaments, specifically we can see keratin. So again, desmosomes are anchoring junctions that will bind adjacent cells together and help form an internal tension reducing network of fibers. We also have gap junctions. So gap junctions are a passageway between two adjacent cells. These allow for passage of small molecules to move directly between neighboring cells. Cells are connected by hollow cylinders of protein and help function in communication between cells, or intercellular communication. So here we see our gap junctions between cells, allowing for passage of ions and small molecules from one cell to the next for intercellular communication. So these channels between the cells are called connexons. Moving on, we're going to talk about the different borders of the cells. So first we have the basal feature. We have the basal lamina. This is located at the boundary between the epithelium and the underlying connective tissue. The basal lamina is a non-cellular supporting sheet between the epithelial tissue and the connective tissue that is deep to it, and consists of proteins that are secreted by epithelial cells. Functions of the basal lamina include acting as a selective filter, will determine what molecules from the capillaries can enter the epithelium, also acts as a sort of scaffolding where regenerating epithelial tissue cells can migrate. So the basal lamina plus the reticular layers of the underlying connective tissue that are deep to it will form the basement membrane. Okay, so the basal lamina is actually part of what forms that basement membrane. So then we have apical surface features. We have structures called microvilli. Microvilli are finger-like projections or extensions of the plasma membrane. They have a core of actin filaments that will stiffen the microvillus. We can find these structures abundant in kidney tubules as well as the small intestine. Basically sites where we want to have maximal absorption and filtration. So microvilli maximize the surface across which small molecules can enter or leave cells. So here we see a picture of the microvilli. We have one microvillus and we have actin filaments here located just deep to that. Another type of apical surface feature are cilia. Cilia are actually whip-like, highly motile extensions of apical surface membranes. They contain a core of microtubules that are held together by cross-linking and radial proteins. We have microtubules that are arranged in pairs called doublets. Movement is generated when adjacent doublets kind of hold on and grip one another along with the motor protein dynein. Cilia originate as microtubules assemble around centrioles. So on the next slide, we can see how these doublets kind of work together along with the motor protein that are dynein arms. And then we can see different phases of the ciliary motion, allowing for propulsion of a layer of mucus that can be found just above the surface of the epithelium. So I know a lot of people get confused between microvilli and cilia. The main difference between microvilli and cilia is basically function. Microvilli increase surface area for absorption of nutrients or transport of ions, also can act and help with filtration. They can also anchor mucus sheets to the epithelial surface, especially in areas where we want that mucus to kind of help protect the epithelial lining. With cilia, however, cilia are made for movement. Cilia will bend and move and are important for the propulsion of mucus and other substances across surfaces. Okay, so just make sure you're aware of the main differences between microvilli and cilia. So now that we're finished with epithelium, we're going to talk about connective tissue. Connective tissue is the most diverse and abundant tissue. You're going to see that again. So again, connective tissue is the most diverse and abundant of all the tissue. And we have main classes of connective tissue. We have 4. We have connective tissue proper, we have cartilage, bone tissue, and blood. And then we're going to go into the different subtypes of these 4 categories of connective tissue. Important functions of connective tissue types include forming the basis of the skeleton, storing and carrying nutrients, surrounding blood vessels and nerves, as well as specific cells that help lead the fight against infection. So just some special characteristics of connective tissue. Contains very few cells, however, are abundant with extracellular matrix. We know that extracellular matrix is composed of ground substance and fibers. Now, extracellular matrix is produced by cells of the connective tissue. And a common embryonic origin is mesenchyme. Now, connective tissues differ in structural properties, and there are differences in the types of cells that make up the different types of connective tissue. Also, differences in composition of extracellular matrix. However, connective tissues all share structural elements, and we know that the loose areolar connective tissue is actually the model type of connective tissue because it will illustrate all the connective tissue features that we can find across the different types of connective tissue. So first we're going to talk about cells of connective tissue. The primary cell type of connective tissue produces the extracellular matrix. So within connective tissue proper, we have fibroblasts. Fibroblasts are the primary cell type of connective tissue proper. They help make protein subunits. In cartilage, the primary cell type are chondroblasts. Chondroblasts secrete matrix in cartilage. Whenever you see the prefix chondro, think cartilage. Osteoblasts. Osteoblasts are the primary cell type and secrete matrix in bone. Whenever you see the prefix osteo, think bone. So notice across the 3 different types of cells, they have this part of the word in common, blasts. So fibroblasts, chondroblasts, osteoblasts. So if you see the word and has the word, the part that contains blasts, know that that means it is the primary cell type depending on the type of tissue. Now, cells in blood are the exception. They do not produce the plasma matrix of blood. And then there are many other important cell types that are found in connective tissue. For example, areolar connective tissue contains fat cells, as well as white blood cells and mast cells. So extracellular matrix that are formed by the primary cells of the tissue is composed of fibers and ground substance. There are three types of fibers found within the extracellular matrix. So fibers function and support and also have unique properties, and we have three types. We have collagen fibers, reticular fibers, and elastic fibers. So collagen fibers are the strongest. They help resist tension. Reticular fibers are bundles of special type of cartilage, and elastic fibers contain elastin, which help with recoil after stretching. Another component of the extracellular matrix is ground substance. Ground substance is produced by the primary cell type of the tissue. So if we have connective tissue proper, fibroblasts will produce ground substance. In cartilage, chondroblasts will produce ground substance. In bone, osteoblasts will produce ground substance. So ground substance is usually a gel-like substance that consists of proteoglycans and glycosaminoglycans. They help cushion and protect body structures. They hold tissue fluid. The only exception is blood, because plasma is not produced by blood cells. Now, if you're going to study and kind of have an idea of the different types of connective tissue, definitely include table 4.2 in your studying. So with this table, we can see the 4 different types of connective tissue, subclasses within these types of connective tissue, the cells that we can find, the components of the matrix, as well as general features of each connective tissue. So we're going to talk about connective tissue proper in just a little bit. We know that connective tissue proper can be broken down into loose connective tissue and dense connective tissue. And we can further subdivide the loose connective tissue into areolar, adipose, and reticular loose connective tissue. For dense connective tissue, we can further divide that into regular dense connective tissue, irregular dense connective tissue, and elastic dense connective tissue. We have the cells that we can find in this type of tissue, components of the matrix. So the matrix of a connective tissue proper will contain all 3 fiber types, specifically collagen, reticular fibers, and elastic fibers. So we know that there are 6 different types of connective tissue and will vary in density as well as in the type of fibers, functions as a binding tissue, and resist mechanical stress, particularly tension. The next type of connective tissue will be cartilage, and we'll go into the different types of cartilage. There's 3. We have hyaline cartilage, elastic cartilage, and fibrocartilage. Chondroblasts, again, look for the prefix chondro. These are the primary cells. They are found in growing cartilage. And then we have mature chondrocytes. These are the components of the matrix. We have ground substance as well as the main fibers include collagen and elastic fibers. General features of our cartilage: resist compression, because of the large amounts of water held in the matrix, also helps to function to cushion and support body structures. Bone tissue, we have different types of bone tissue: compact bone and spongy bone. Again, the primary cells are the osteoblasts, and we also have mature osteocytes. Matrix is made up of ground substance that is calcified, and then fibers are collagen fibers. We know that bone tissue is a hard tissue that will resist both compressive and tensive forces and helps function in support. The 4th class of connective tissue is blood. We know that blood cell formation and differentiation is very complex, and we'll talk more about blood in chapter 18. We have different types of cells within blood. We have our erythrocytes, or our red blood cells, leukocytes, white blood cells, and our platelets. The matrix is composed of plasma. We don't have any fibers in blood. General characteristics of blood: it is a fluid tissue, helps to function by carrying oxygen and carbon dioxide, as well as nutrients, waste products of metabolism, and other substances such as hormones. So I would definitely use this table when you're studying the 4 different types of connective tissue. So first we're going to go into connective tissue proper. So the 2 subclasses of connective tissue proper include loose connective tissue as well as dense connective tissue. Under loose connective tissue we have loose areolar connective tissue, we have adipose and reticular loose connective tissue. For dense connective tissue we have dense irregular, dense regular, and elastic connective tissue. So embryonic connective tissue is called mesenchyme. Mesenchyme has a gel-like ground substance, and these cells are star-shaped mesenchymal cells. All connective tissue arise from mesenchyme in the embryo. So here we can actually see a picture of embryonic connective tissue or mesenchyme. We can see the star-shaped mesenchymal cells surrounded by ground substance, and then we have some sparse fibers within the matrix. So we know that mesenchyme gives rise to all other connective tissue types, and it's primarily found in the embryo. So connective tissue proper, we will first really talk about areolar connective tissue. Areolar connective tissue will help you understand the properties of many of the other connective tissue types. Areolar connective tissue has many structural components common to other connective tissues. That's why it's called the model connective tissue, meaning it has the ground substance or the matrix that is made up of the ground substance as well as all 3 fiber types. It has the primary cells. So again, it is known as the model connective tissue. So extracellular matrix being composed of ground substance, which is that gel-like substance, and the 3 different fibers, the collagen fibers, elastic fibers, and reticular fibers. We can see the fibroblasts, which help secrete the matrix. And then we can see mast cells as well as other cells. So again, areolar connective tissue is the model tissue type. This areolar connective tissue actually underlies epithelial tissue. It surrounds small nerves and blood vessels, has structures and functions that are shared by other connective tissues. This areolar connective tissue borders all other tissues in the body. Major functions of connective tissue. We know that the structure of areolar connective tissue will reflect what it does or reflect its function. It supports and binds other tissues. It also holds body fluids. For example, within the interstitium, we have interstitial fluid, and then we have lymph. It helps defend the body against infection. We have specific cells that help defend against infection. Also important for storing nutrients as fat. So we talked about the fibers within connective tissue. We have fibers that provide support, and there are 3 types of protein fibers in the extracellular matrix. We have collagen fibers, reticular fibers, and elastic fibers. Again, our primary cell, which is the fibroblasts, produce these fibers that are contained in the extracellular matrix. So within areolar connective tissue, the gel-like matrix will contain all 3 tissue types. We also have cells of the areolar connective tissue, our primary cell, which is the fibroblasts. There are macrophages, mast cells, and white blood cells. Function of the areolar connective tissue is to wrap and cushion organs, also hold and convey tissue fluid. This is known as interstitial fluid. Also has an important role in inflammation. Areolar connective tissue is actually widely distributed underneath epithelia, helps package organs, and also helps surround capillaries. So here is a picture of loose areolar connective tissue. You will see this picture again because it's one of the connective tissue that you are responsible for. Again, it is the model tissue because it has structures that can be found in all types of connective tissue. So we have our different fibers. We have our elastic fibers, our collagen fibers, as well as our reticular fibers. We have our fibroblasts, macrophages, mast cells, and some white blood cells. Again, its function is to wrap and cushion organs. We have macrophages that will help phagocytize bacteria, meaning it engulfs bacteria, sort of like a Pac-Man cell. Also plays an important role in inflammation and helps hold and convey tissue fluid. Again, it is very widely distributed under the epithelia of the body and helps form that lamina propria of mucous membranes. Also packages organs and surrounds capillaries. So here we can see just a rough drawing of the epithelium and forming that lamina propria of mucous membranes. Now, the tissue fluid is also known as interstitial fluid. It is the watery fluid occupying the extracellular matrix. This tissue fluid actually derives from blood or comes from blood. The ground substance is that viscous, spongy part of the extracellular matrix. Consists of sugar and protein molecules and is made and secreted by fibroblasts. Again, fibroblasts being that primary cell within connective tissue. So fibroblasts are not only important in the production of the 3 different fibers, but they also produce the ground substance of the extracellular fluid or extracellular matrix. So another characteristic of areolar connective tissue, it is the main battlefield in the fight against infection. So we have our defender cells, or defenders that gather at the infection sites. We have our macrophages, plasma cells, mast cells, and white blood cells. White blood cells being our neutrophils, our lymphocytes, and eosinophils. So that's it for part 1 on the chapter of tissues. This is actually a pretty long chapter, so I'm going to kind of divide it up for you guys so that you don't have one long video to listen to. So this is the end of part 1. Stay tuned for part 2.